the cloud, and we're also streaming it live on SciStarter's Facebook, in part because I joined like 20 different ASU groups this week to help promote this. So I want to make sure that we share it with as many people as possible so we can reach them with the citizen science message. So because of this recording, if you're with us on Zoom, we may read out whatever you put in the chat. It'll be part of our record that we share with folks. But we don't want that to discourage you from engaging with us. We really want to see some activity in the chat. We want to be responsive to what you want to hear about, because this webinar is really for you. It's for Arizona State University and the wider community. And we're just so excited to be celebrating the upcoming Citizen Science Month with you. So I'm just going to get the Facebook live stream going. We'll also post it on YouTube as well, because I know sometimes Facebook isn't always friendly with everybody. So we will find a way for you to get this information during this event and afterward. So as we get that set up, feel free to say where you're calling in from. If you have something you're already excited to learn about, tell us that. We want to be responsive to what you want to hear about. And the Facebook is now live. I'm going to put the link in the chat just so you all have it during this event and after the fact. All right. So let's get started. My name is Caroline Nickerson. I'm with the SciStarter team. I'm going to be your host today. I'm so excited to have so many special guests from the ASU community here to tell you about the field of citizen science, which is public engagement and real scientific research. And if you don't know what citizen science is, that's okay. You don't have to know. We're going to tell you all about it. If you know everything there is to know about citizen science, hopefully you'll learn something new during this event. And also, hopefully you'll rise to the call to action and make April your month to engage. We have a special microsite we made just for you at SciStarter.org/ASU-SitSciMonth 2021. So if you want to minimize me and if you want to go ahead and pull that up in another window, feel free to do that. I won't stop you because we made this microsite for you and we want you to explore it. For those of you who are watching either via the Facebook live stream or you're watching the recording and not joining us in Zoom, don't be afraid to give us a shout either on that forum on that SciStarter microsite or via email at info@scistarter.org. We really want to be responsive to you during your Citizen Science Month experience. So to get us going, tell us about yourself. I know some of you already started doing that, but tell us where you're calling in from. If you've ever participated in citizen science before, if you've ever done a project, and why you signed up for this webinar, just because we're curious. There are no wrong answers here, and feel free to let us know in the chat. I'm going to pull up the chat window so I can see what people said. Someone said, I'm from Berkeley and my son is at ASU as a freshman. I saw pollinators in the topic and we keep beehives at home on the side of our house. So I wanted to see what more is going to be part of this topic. That is awesome. I'm so glad we have a Sun Devil mom in the chat with us. And someone asked, can you put the link in the chat? Yes, I can. We will put SciStarter.org/ASU-SitSciMonth 2021. There we go. So feel free to introduce yourself. Feel free to tell us why you're here. And I noticed some of you are interested in pollinators. We have special guests who will tell you all about pollinators. So to get started, we always like to start with an introduction to what citizen science is. So millions of people enjoy science and nature. You all probably count yourself in this number. Maybe you're really excited by bees, pollinators, and flowers. Maybe you are intrigued about how the brain works and you want to understand it a little bit better. Maybe you're just really into stargazing and you love to go outside and ponder the night sky. You're in good company. There are millions of people just like you. And the good news is thousands of scientists need volunteers. There's research out there that wouldn't happen without you. Scientists don't have enough eyes, ears, and perspectives to solve every problem there is to solve, to answer every question there is to answer. To understand the world, they need the volunteers just like you to go out there and find data, analyze data, basically to turn their curiosity into impact to move science forward. And we're going to describe how you can do that with 3 different projects today. However, sometimes these folks can't find each other. The scientists who need volunteers can't find the people like you who are so passionate and ready to get started. That's where SciStarter comes in. We connect them. SciStarter is a way for you to discover citizen science. What is citizen science? Citizen science is a collaboration between scientists and those of us who are curious, concerned, and motivated to make a difference. Basically, citizen science is whenever someone is meaningfully part of the scientific process. Citizen scientists often join ongoing research projects just as a hobby, just because they want to help and they contribute to that mission. Most often, citizen scientists are volunteers, and anyone can be a citizen scientist. Everybody from Girl Scouts to retirees. And to hammer that home, we have a video about SciStarter, which is one of the partners putting on this event with ASU for Sci Month. So I'm going to stop sharing my screen really quick because I want to make sure I'm sharing my sound, and I am. And we're going to go ahead and play the video.
Citizen science can change the world. These programs mobilize people from all walks of life to contribute to scientific research, from teachers to artists, Girl Scouts to business professionals, students to retirees. Citizen science taps into the power of the people, where they are. Scientific data gathered by individuals and communities gives concerned or curious people, decision makers, and scientists access to more information than they could possibly gather on their own. Your contributions lead to breakthrough research, new approaches, and expedited scientific discoveries. So how can you start to make a difference? SciStarter makes it easy to find the perfect project for you. With a database of more than 3,000 citizen science projects to choose from, you can search by topic, location, age level, and more. Anything that sparks your curiosity is a mere click away. SciStarter can help you take valuable actions to contribute to the projects you're most passionate about. Their tools database provides you with exactly what you need to get started. And with events held across the nation throughout the year, SciStarter brings fun citizen science opportunities directly to you. Visit the SciStarter website to begin exploring the possibilities of scientific participation and create a personal account to receive project suggestions tailored especially for you. Together with SciStarter, we can all play a part in scientific research that can change the world for the better.
And there you have it. That's citizen science and SciStarter in a nutshell. And I'm so delighted to announce that we have the founder of SciStarter on the line, Darlene Cavalier, who's also a professor of practice at Arizona State University School for the Future of Innovation and Society. Darlene, could you say a few words for our ASU Sit Sci Month kickoff?
I am, first of all, sorry I'm not on camera, but it's great to see all of your faces and have an opportunity to tune in all the way from the other side of the country in Long Beach Island, New Jersey. But we're very, very excited to have an opportunity to involve students and faculty and others, visitors, others around campus to help some ASU researchers, like I said in the chat, answer questions they might not be able to answer without the help of the citizen scientists. Thanks for this.
Yeah, and thank you to you, Darlene. Darlene and I right now are working with the Osher Lifelong Learning Institute of Arizona State University to activate retirees in the community to be citizen science facilitators. So you can be any age, any walk of life, you can do citizen science, and the Sun Devils are definitely coming in strong for Citizen Science Month, which is so exciting. So I wanted to include this cartoon. I think it's very funny, and it really underscores what we're basically undertaking a group project together. I think you'll be delightfully surprised by the quality of my work on this assignment. I crowdsourced it. I think this really underscores the fact that we have a call to action for the Arizona State community and for all the people in Arizona in general. If you all activate during Sit Sci Month and contribute to these projects that you'll hear about today, you can accomplish so much more than you would by yourselves. There are insights about pollinators and native plants that we might have because of the work of you all. So this is your call to action. Everything is on this page. We already mentioned it and put it in the chat. If you're watching on Facebook, feel free to type in this URL if you're watching the recording. Here it is. I'll also put it in the chat again for our Zoom attendees. this is kind of your one stop to engage with ASU Citizen Science Month activities. On this page, you'll find the featured projects, you'll find a tutorial, you'll find the opportunity to sign up for kits from ASU Destination West. You'll find information about the seed library and so much more. And you'll also find other RSVPs for different events offered by the same groups during Citizen Science Month. We also wanted to kind of kick off this event with these pictures. You may recognize this face. This is Sparky the Sun Devil. And he is also celebrating Citizen Science Month. As you can see, he's monitoring native plants in his community, and he had a QR code to engage people in Citizen Science Month online that links to the webpage that you all have access to as well. So here, I'm going to pass the mic to Carolyn Starr from ASU Destination West, who's going to talk about an opportunity she put together for the month, which is a Citizen Science Kit. Carolyn?
Thank you, Caroline. So yeah, we are super excited. We are offering free kits to the first 50 people who want to pick them up. We have created in this kit just a little bit of different things. We have sunscreen because you're gonna be out in the sun, a little first aid kit so you don't get poked with a sticky branch. We're fortunate to be able to add the field guide to Citizen Science book, a flash drive with a bunch of other resources on it so we don't waste paper, but you can look at those and be able to download some more information if you want that. Bookmarks, and then you're going to get a seed packet that has milkweed seeds in it, which they'll talk about a little bit later for one of the resources for creating your own area to bring pollinators, and also a word search. So there's a lot of little goodies in there. There is a link where you can go. It's also on the SciStarter page, but you can go to this link and it will let you sign up for the kit, and then we'll give you email directions on where to pick it up at the ASU West campus. So we're super excited about that, and we hope we find 50 homes for them. Thanks.
Thanks, Carolyn, and thank you for making this kit and giving people the tools to do citizen science. Now we have a video. This was created by Alice, who's on the line, and we're going to play it before we go into her portion of the presentation.
Hey everyone, this is Alice with ASU's Center for Biodiversity Outcomes. Did you know that pollinators are vital to healthy ecosystems? Birds, bats, butterflies, bees, and many other species are necessary for the health of flowering plants, food crops, and the global economy. We literally could not live without them. This April, join us in documenting pollinators and flowering plants on ASU's campuses. It's super easy to participate. Let's get started. All you need is a smartphone to take pictures of pollinators and flowering plants, the iNaturalist app, and a SciStarter account. Head over to SciStarter.org to set up that account, get a detailed project tutorial, and start snapping pics.
All right, and now you're going to hear from Alice about the Center for Biodiversity Outcomes and why they're participating in this effort. Alice?
Thank you so much, Caroline. As that video told you, my name is Alice Fletcher. I'm a project manager with the Center for Biodiversity Outcomes at Arizona State University. The center is a partnership between the Global Institute of Sustainability and Innovation and the School of Life Sciences. Next slide, please. So what is biodiversity and why does it matter? The word comes from the Greek root bio, meaning life, and the Latin root diversitas, meaning variety. It's the variety of life, and that encompasses ecosystems, species, communities, processes, and genetic diversity. It's the web of life that's all around us on our planet, and we could not live without it. We rely on it for food, shelter, medicine, clean air, and water. Next slide, please. So this project is intended to engage citizen scientists in collecting data on pollinator and plant species on and around ASU's Phoenix area campuses. And the goal is for us to better understand the distribution of those pollinators and plants, and to understand how we can engage the public in our research efforts. Next slide. We have three specific questions that we're looking at. The first is, do pollinator species and species densities vary with location? The Phoenix area ASU campuses are pretty diverse in their locations and in the kind of vegetation you see on campus. And we want to know what trends can we observe across those campuses. Next slide. Our second question is, do pollinator species and species densities vary with plant type, specifically looking at native versus non-native plants? Some pollinators are very specific in the types of plants that they can make use of, like the bat picture you see here. And some are much more agnostic. They can access a wide variety of plants. So we want to look at, are there more pollinators where there's a higher density of native plants, or is it more homogeneous than that? Next slide, please. And our last question, and where you really come into this, is how do data collected by citizen scientists compare to data collected by professional scientists? We want to do a better job of engaging the public and engaging citizen scientists in our research, and we need to know how to do that better. So I hope you will join us and help us out.
All right. Thank you, Alice, for that call to action. Now we have a presentation that was created by a student at ASU, Jared, an undergraduate student. And I am just so impressed by his tech skills. He has it all embedded into this PowerPoint. So let's hear what he has.
Hello, everyone. My name is Jared, and I'm a undergraduate researcher in the Conservation Innovation Lab at ASU and working with the Center for Biodiversity Outcomes in developing this project. This presentation is a short overview of the importance of pollinators, pollination, and native flowering plants, and this pertains to two of the projects in Citizen Science Month, Pollinators of Arizona State University and Flowering Plants of Arizona State University. So what are pollinators and why are they important? Pollinators include bees, birds such as hummingbirds, bats such as the lesser long-nosed bat, butterflies, beetles, and small mammals that provide an important ecosystem service of pollinating plants. They either collect pollen as a food source or unintentionally carry pollen on their bodies from one flower to the next as they forage for nectar. This is a free ecosystem service, and it's responsible for 1 out of every 3 bites of food you eat. But they are declining due to climate change, chemical usage with pesticides, diseases, and loss of habitat and food. Pollinators contribute roughly $217 billion to the global economy every year by indirectly benefiting the agricultural sector. It would cost less for us to preemptively act to conserve them than it would cost to lose their ecosystem services and fill the void left behind. So what is pollination? Pollination occurs when pollen is moved from the male part of a flower, the stamen, to the female part of the flower, the stigma, of the same flower or another flower of the same species. This fertilizes the flower and produces fruits and seeds. Some plants rely on the wind to transport pollen, but others rely on pollinators to unintentionally move the pollen from flower to flower. An example of this are butterflies. As they feed on nectar, they get covered in pollen, and as they flit from flower to flower, they transport it to other flowers as they look for more nectar. The fruits and seeds that result from pollination provide food for other animals such as birds and squirrels, and the seeds that fall to the ground will grow and continue the cycle. When thinking of pollinators, usually one species comes to mind for most people, and that's bees. And there's good reason for this. Bees are the most effective pollinators, and they are highly organized within their social structure. There's an estimated 20,000 different species of bees in the world, with 4,000 species found in the United States. But Arizona actually has the highest native bee diversity in the US, with 540 bee species found in southeastern Arizona alone. An estimated 35% of the bee species are oligoletic, meaning they have preference for certain flowers, which limits their habitat range. An example of how effective bee pollination is, in 2010, pollination by honeybees in the United States contributed $19 billion in harvested crops, and this is a free service for the farmers. But not to discount the work by others, butterflies, bats, birds, and other pollinators play a crucial role in ecosystem services as well. The Southwest has 18 species of hummingbirds and 3 species of nectar-eating bats. The lesser long-nosed bat was a previously endangered species that plays a vital role in a desert ecosystem and for pollinating cacti. So when there are numerous factors threatening pollinators, the main threat is habitat loss, degradation, and fragmentation, where human development, such as constructing roadways and parking garages, removing habitat and planting non-native species in gardens, greatly reduces preferred native food sources. The use of pesticides to destroy pests has an inadvertent effect of harming pollinators. Climate change can displace pollinators, and parasites and diseases reduce pollinator populations. As pesticides are used to target unwanted pests and fungi, they have the inadvertent effect of harming pollinators as well. Pesticides can remain in an ecosystem and have a negative impact for a long period of time. Insecticides that are applied to seeds can later contaminate the pollen grains in the mature plants that serve as a food source. If the usage of pesticides does not kill the pollinator, it might harm its sense of navigation and its ability to forage. With the threat of climate change, the preferred types of flowering plants for pollinators in a region might disperse to higher elevations and further north. This dispersal could be aided by animals such as squirrels and birds that will drop the seeds off as they move northward. This would lead to a change in the type and distribution of pollinators, as well as potentially displacing the pollinators already inhabiting a region. Introduced non-native parasites and diseases harm native pollinator populations. An example of this is the invasive aurora mite. It originates from Asia and it's a threat to honeybees on a global scale. Acting much like a tick, as seen in the picture on the right, it infiltrates the hive by attaching itself to a bee and grows in population within the hive. This mite can be a vector for diseases, especially for the western honeybee, which has no immune defense against the mite. A native species can be devastated by a parasite and disease that's invasive, just because it has no encounter in the past with it and hasn't built an immune defense against it. The introduction of non-native plants to an ecosystem can lead to a decline in the native plants as they are outcompeted for resources. Not to be confused with invasive plants that can be devastating for an ecosystem, non-native plants are often bought at local gardening centers and they can be found on campus. These non-native plants can take pollinator attention away from the native plants, even though the non-native species might lack in nutrition. Pollinators and native plants have co-evolved to best suit each other's needs. The introduction of non-native plants is a disruption to the mutualistic relationship. As mentioned in a previous slide, some bee species are oligoletic and have preference for certain flowers in a limited habitat range. The various threats mentioned that are harming pollinators and the use of non-native plants could have a significant impact on the specialized bee populations within these small ranges. Ways in which you can help out at home. You can plant a native flower garden, provide nesting habitat in your backyard, use and call for pollinator-friendly alternatives to pesticides, or support nature corridors and refuges. When planting a garden at home, you want to choose native plants that will attract native pollinators and serve as larval hosts for some pollinator species. You want to plant in clumps to maximize visibility and attractiveness instead of having flowers that are very dispersed and distanced away from each other. You want to plant with a variety of colors and shapes to attract different species of pollinators. Add a native milkweed plant to attract butterflies, and reference a guide like the one found on the next slide that will help you find the best suitable flowers for pollinators in your area. When planting a garden at home, you can reference a guide such as this one, the Pollinator Syndromes Guide. This is provided by the North American Pollinator Protection Campaign, and they have a lot of additional resources on their site that are very helpful. But this guide references the flower characteristics that are most appealing to certain pollinators. So if you were trying to attract, say, a bat, like the lesser long-nosed bat, to your garden, you could go to the type of pollinator, find bat, and you'd see that it likes white, green, or purple colored flowers, strong and musty odors that are emitted at night, abundant nectar, ample pollen, and bowl-shaped flowers that are closed during the day. These characteristics are why they are closely associated with pollinating the saguaro cacti. Another way in which you can help out at home is to provide nesting habitat. So pollinators have varying preferences in nesting habitat. You'll find that hummingbirds build nests using moss, lichens, and spider webs and tend to camouflage their nests in trees and shrubs. Butterflies will lay their eggs on plants like a milkweed plant. Then you'll have the ground nesting species that look for an undisturbed and well-drained patch of dirt that faces south. Make sure there's no sprinkler systems that are nearby. And then some species nest in old wood, so consider placing a log in the garden as maybe a backdrop. And lastly, you could always avoid using pesticides. So you could remove pests by hand. You could consider promoting native predators that will eat pests in the garden. An example of this, and on a large-scale application, would be in the winter garden region of Texas, where the bats flying over the crops save the farmers around 1.7 million a year by eating the pests harming their crop yield. This is another free ecosystem service, and as a farmer, I would consider placing bat houses around my crops. But if you must use a pesticide, apply it in a small quantity and in a localized region, and choose a time of day where the pollinators are the least active and there's little to no wind. This way, the pesticide will not spread to nearby flowers or plants. So now that we've gone through a brief overview of pollinators and the importance of habitat, the question is, how can you help as a citizen scientist? You can participate by signing up for Citizen Science Month at SciStarter and iNaturalist. Grab your mask, camera, and head to your nearest campus and take photographs of at least 6 different pollinator species and at least 6 different flowering plant species to then upload to their project profiles on iNaturalist. This information will be used to create an inventory of pollinators and flowers on campus and promote conservation efforts, such as providing better habitat. Here are 2 common pollinators that you'll most likely come across. The first is the western honeybee, which is quite common. It's very prevalent in Arizona. And the queen butterfly, which I found was more prevalent on the Polytechnic campus because that campus has more milkweed planted compared to the Tempe campus. Two uncommon pollinators would be the lesser long-nosed bat and the white-lined sphinx moth. It would be surprising if someone got pictures of these because they pollinate at night. But that being said, if you're a photographer and are up for a challenge, we would love a picture of the lesser long-nosed bat. And 2 common flowering plants would be the saguaro cactus and desert marigold. And if you look at the pollinator syndromes reference guide, you will find that the saguaro cactus will attract nectar-eating bats, and the desert marigold would be popular with bees.
All right, and there you have it. That was Jared's prepared presentation. I'm gonna exit my screen there, and we're gonna go back to the other programming. So Jared educated us about flowers and pollinators that you might discover at ASU. And you can find these projects and how to participate on our SciStarter page. Before we do that, you're going to hear about another resource available on the SciStarter page. This not available on the SciStarter page, but more information about it's on the SciStarter page, the seed library. So I'm going to pass the mic to Christina from ASU Library.
Hi, so the seed library is located at the design library on the Tempe campus, which is in the design north building. So the seed library is for ASU ID card holders only. But if you're not an ASU ID card holder, a lot of public libraries have it. So I searched some of the public libraries, and Mesa is one of the public libraries that have seed libraries. So if you are interested and you're not a part of the ASU community, you can go and check out your local library as well. So the seed library, we have heirloom seeds, and we also have desert flowers and local flowers as well in our seed library. So they're native flowers, and we also have like a lot of the like poppies and other seeds that grow in the springtime. So we also accept donations as well. And if you want to learn more, you can always go to our LibGuide as well, which is at libguides.asu.edu/seed library to find out more information. We have on there information about when to plant seasonal seeds and stuff like that.
Awesome, good stuff. Thank you, Christina. And we wanted to showcase this because there are so many different ways that you can become part of nature and really celebrate the nature of Arizona, especially. And that's a great example. But we have a third featured project that's available on that page. It's the Stall Catchers project, and you can do this one from anywhere. So the projects you've heard about so far have been really focused on Arizona and the ASU, especially in the community surrounding it. But Stall Catchers, there are people in Nigeria who are playing it. There are people in Asia who are playing it. And of course, I play it from my own home as well. So I am going to actually demo it for you today because we have time. So if you go to SciStarter.org/ASU-SitSci Month 2021, you make it here. You can make a SciStarter account on the left side of the page. So I'm going to log out just so I see what you all see. So this shows up if you don't have a SciStarter account already, and you can make your SciStarter account. Please make it with, if you have an ASU email, make it with your ASU email. If you don't have an ASU email, I don't, for example, just use the email you use the most, because this is really valuable for tracking for us. For the projects on this page, they're SciStarter affiliates. So you're able to keep track of the number and frequency of your contribution to different projects and your SciStarter dashboard, as long as you follow the instructions on each page. So the two projects Jared described, they're right here for you to discover. You can read the steps. If it was a little too fast in this presentation, don't worry. You can read the steps and get started. And then you have a participate button you can click to go right where you need to go. But the Stall Catchers project, that's our third project here. There's a video from the project leader, Pietro Michelucci, talking about why he started Stall Catchers. A little bit of background info about how Stall Catchers accelerates Alzheimer's research, and then step-by-step instructions, especially about how to make sure your contributions track. The main thing you need to make sure of is that you use the same email that you use to make your SciStarter account to make your stall catchers account. And when you click the link right here to participate, once you make your stall catchers account, you'll automatically be on the Arizona State University stall catchers team. As you can see, I'm on the Arizona State University team because I consider myself part of the wider ASU community by being part of this effort. It's Citizen Science Month is something to celebrate, and I'm excited for my stall catchers points to go toward ASU. So you might be wondering, what on earth am I looking at? This is a video of a mouse's brain. These white things here are blood vessels. What you're trying to do with stall catchers is, hence the name, you're trying to catch the stalls. These researchers from Cornell University have pinpointed their research on a question about blood flow in Alzheimer's disease. And believe it or not, they're able to go through certain data sets and answer certain questions about Alzheimer's based on your annotations of these videos, whether or not a particular blood vessel is stalled or flowing. You know a blood vessel is stalled when there's a persistent black spot in the flow of white that's not moving. And you're supposed to look at the blood vessel, the flow of white, that most closely matches the circled area. So I'm going to do a few to get us started, and then I'm going to have you all vote in the chat. So I'm slowing it down a little bit. You can have the video play on a loop, but you can also use the slider. And I think I spotted a stall. I see a persistent black spot in the flow of white that's not moving. So I'm going to say stalled, and then I'm going to click where it's stalled. And I'm correct. It is stalled. I'm getting pretty good at this. The cool thing about stall catchers is the folks who also play chime in. They can give their analysis. If there's an expert and they really think it's stalled, they can explain why. And it looks like my answers agree with the rest of the community. About 2,000, more than 2,000 other people also thought this blood vessel was stalled. And you might be wondering, oh my gosh, if I'm actually making these annotations, could I mess up the research? The answer is no. The stall catchers team has planned for that. So more than one person, a lot of different people, as you saw by those thousands of folks who'd also analyzed this video, make these annotations. And the project is designed in such a way that if your answers agree with the crowd most of the time, your annotations are ranked a little bit more highly. And as you progress, it might trust you more and it might assign you more points. That's what part of makes it fun. That's part of what makes this project fun. It's gamified, so you get points for how much research you do. And you might also be wondering, couldn't computers do this? The answer is not yet. It still needs the human eye to analyze these stalls, but how the stall catchers team has utilized computers is making sure to rank your contributions depending on how often you agree with the crowd or not. And that's the beauty of this. I can trust in my fellow catchers, so even if I'm not always right, the crowd's always right. And the best way to learn this game, I always tell folks this, you just got to do it. The more you annotate, the more videos you go through, the better you'll get. And I hope you have fun with it too. I really love seeing my scores go up and my points go up. And I also like the fact that I'm part of the ASU team, and who knows, maybe we'll score the most points during Citizen Science Month this April. So let's do another video. All right, we're watching this on a loop. I need to look at the blood vessel that most closely matches this circled area, which I think is that flow of white there. And it looks like I do see black spots in the flow of white that looks like they're moving. So I'm gonna say it's flowing. Let's see if my answer agreed with the crowd. It did. And the expert answer, so the person, probably somebody who's on the leaderboard who has some of the most points of all time, they say, if you see a segment stall for only a few frames, the segment must be stalled for the entire length of the video for it to be considered stalled. Good to know. All right, we got the circled area here. We're looking at that blood vessel in the circled area. We're looking at the flow of white that most closely matches that shape. And we're trying to see if there are any persistent black spots in that flow of white that don't move. And I have an idea of what I think the answer is, but I want you all to vote. What do you think in the chat? Is this blood vessel stalled or is it flowing? And you know what? I'm going to call on one of my friends here. Carolyn, what do you think? Is this blood vessel stalled or flowing? Carolyn says question mark, question mark. That's okay. You don't have to know quite yet.
I put stalled up above, so I was thinking it was stalled, but I'm not sure. I've tried this game. I'm not very good at it yet.
Hey, it takes time, day by day. And then it looks like Alice says flowing. So we have a stalled and we have a flowing. Do we have a tiebreaker vote? Maybe Darlene, will you be our tiebreaker? We'll see what she puts in the chat. She says flowing. All right, we're going to go with flowing. Majority rules. And we're correct. It's flowing. Expert answer, vessel is flowing. That's not the most helpful answer, but that's okay. I happen to agree with them, so we'll keep going. All right, let's do one more. So we have a circled area here. We're looking at the blood vessel that most closely matches the circled area, which I believe is that blood vessel. And one thing to keep in mind, you might be wondering, why is this video so bad? That's just the nature of research. Some videos are good, some videos are bad. It's the same thing folks would be dealing with in the lab. All right, what do we think? Stalled or flowing? This isn't the best video, so I'm just gonna say flowing. All right, you all get one more. And Jared said flowing too, so he agreed with me. This is our last one. So we're looking at the blood vessel in that circled area. So I think it's that shape. And we're trying to see if in the flow of white there, if there are any persistent black spots that don't move. What do you all think? Stalled or flowing? Vote in the chat. And for those of you watching on Facebook, I'm monitoring that as well, so you can vote in the comments, so to speak. All right. We have some folks tuning in on Facebook, but I think they're a little shy. So let's see what our friends on Zoom say. I gotta say flowing for this one, I think. Does anyone disagree? All right, the people also agree with flowing. Good to know. And I have progressed far enough that I'm seeing new stuff. That's how you know I played a lot of stall catchers, and they're asking me to comment. I might come back later. So this is our ASU team. There are quite a few different folks on the team. The team score is pretty high, which is exciting. And we're not in a league at the moment, but this team's been around all the way since 2019. So you're joining a team with some storied history here. Very, very cool. And one nice thing about stall catchers is you can also learn a lot while you do it. So while you're classifying these different data sets, the stall catchers team will actually tell you the type of research you're doing. You don't have to know every single detail, right, involved with the research to follow the protocols, follow the instructions, make a difference. But if you're curious, you can, for example, this is about a certain gene mutation. That's the particular data set we're analyzing right now, and it's a new one. They update their blog pretty frequently, which I think is phenomenal. One thing that came up pretty recently is they published a study in one of the most important scientific journals. So the researchers behind stall catchers at the Human Computation Institute in Cornell University, they published it. And this journal, they cited the citizen scientists, so volunteers just like you, as co-authors, which I think is phenomenal and really exciting. So by joining this project, you all are joining one of my favorite citizen science efforts, and you're a meaningful part of the scientific enterprise. So that was stall catchers, and you can discover that on that SciStarter.org ASU microsite. So just to review, things you can find on SciStarter.org/ASU-SitSci Month 2021. You can find information about how to reserve a kit from our friend Carolyn at ASU West. You can find the sign up there, so make sure you sign up for your kit. And she's having people pick up kits as early as tomorrow, so get there before it's gone for sure. And Carolyn also mentioned, you know, in addition to the bookmarks in the book and all the resources she has in there, there's some milkweed seeds too. so you can take action on some of the issues Jared identified in his presentation. There's also, we're adding this ASAP, but Jared made a word search and then Carolyn's team made some beautiful imagery behind it. You can warm up with a citizen science word search. I really like word searches, so I'm definitely printing this out after the presentation and participating. So definitely get started with your Citizen Science Month word search on the SciStarter.org/ASU-SitSciMonth 2021 microsite. And then a link to find more details about the ASU seed library that Christina told you about. That's also on SciStarter.org/ASU-SitSciMonth 2021. And our 3 featured projects, you can start today with Pollinators of ASU, Flowering Plants of ASU. The call to action is for folks in Arizona to really go outside and start documenting the pollinators and flowers that they see. But you can also do stall catchers from anywhere. So I will definitely be participating. And I know the ASU stall catchers team, perhaps you all could dominate this year. Give the Los Angeles Public Library, our reigning champs, you can give them a run for their money. And we have another event on April 1st. So if your interest is really piqued by this presentation, and if you have additional questions, we're going to be reviewing these efforts again on April 1st for our official kickoff. This webinar was just for the really eager folks who wanted to get started right away. And another thing we're doing on April 1st that's a little bit different from this one, I've heard a rumor that there's going to be video footage of Sparky the Sun Devil participating and counting the pollinators and local plants on ASU campus. So you don't want to miss that. You don't want to see that video. So if you want to register for this second event that covers the same topics that we covered today with a little bit of extra info for the kickoff, go to SciStarter.org/ASU-SitSciMonth 2021. We're excited to participate with you all April. And if you are on social media, feel free to use the hashtag ASU-SitSciMonth. I'll be monitoring it. I know some of my friends on the line will be as well. We really want to celebrate your efforts and the cool stuff you're doing, so don't be afraid to use that hashtag so we can help spread the word. And that's everything we had for you today. I'm going to stop sharing there and see if my co-presenters have anything else they want to share before we all go. And if you all have any questions, this is your last chance to get them in the chat. And our Facebook friends, you can also chime in with questions too. I know a few people are watching live on Facebook.
You did great today, Caroline. Thanks for hosting this.
Oh, you did great today, Carolyn. Thank you so much. This was fun. And oh my gosh, Hillary said in the chat, have a beautiful day. All right. Well, if my co-presenters, another thing in the chat, informative, this will be a useful resource on the website for visitors. That's right. That's an important thing to mention. So for those of you who are watching, if you want to go back and review anything from any of the presentations, it'll be up on the microsite, SciStarter.org/ASU-SitSciMonth, and you can watch it again. And if you have additional questions, don't be afraid to join us again on April 1st and ask them. April will be a buzzy month. Yes, it will. It really will. And if you have some puns, use that ASU SitSciMonth hashtag, tweet them at us. Tweet them at SciStarter, tweet them at ASU. All right. Have a good night, everybody. Good afternoon, I mean. Have a good afternoon. Bye, everybody.